Hello, everybody. Thank you for tuning in again. Today we're going to talk about protein synthesis. So far we've talked about DNA structure, DNA replication, that central dogma of biology going from DNA to RNA to protein. And then we also talked about those different types of RNA, whether it's the mRNA, the messenger, the tRNA, the transfer RNA, which brings in those amino acids, or the rRNA, which is just our ribosomal RNA. Today we're going to talk about that whole step, that central dogma, pretty much protein synthesis, going from our DNA to our RNA to our protein, and kind of seeing how it happens. So to get started, proteins. Protein synthesis is the process all cells use to make proteins. Again, proteins are our most diverse macromolecule. They do a ton of different things, including giving us our traits that we have. Now, again, that central dogma, going in the steps of protein synthesis, we have our DNA here in our nucleus. We go through transcription to create our mRNA, our messenger, and then we go out into the cytoplasm for translation, which is going from RNA to that protein aspect. Now, it's significant here because our DNA, we want to keep that in the nucleus. It's protected in there. We don't want it to leave the nucleus at all. So we create this mRNA in order to allow it to leave the nucleus, to go out to the cytoplasm where it can be translated. So again, proteins are responsible for all cell structures and functions. DNA has the instructions to make those proteins, and proteins are made at the ribosome, such as in the rough ER. Now, again, as I said, that nucleus is housing that DNA. We don't want it to get out because if it got out of the nucleus, then there might be some things in the cytoplasm that would want to break it down and destroy it, and we really don't want our DNA to be destroyed. It's not good. So we create this mRNA that goes out, goes through translation, and then that actually gets broken down as well. So protein synthesis steps. Our first one is our transcription. Now, again, transcription, DNA and RNA speak the same language. They speak the nucleic acid language. So we don't need to translate anything. It's just going from one nucleic acid to another nucleic acid. We're writing down, we're scribing or scripting the message. So DNA is copied to mRNA. Now, what you'll notice between DNA and RNA is that RNA is only single-stranded here. Move my box. There we go. RNA is only single-stranded. So again, we're only using one strand of this DNA to create this RNA molecule. However, keep in mind DNA is that runs opposite. So this top here going down, it's going to be the same as this one here going up. It just runs in opposite direction. So our RNA molecule is only going to be our one strand based on that one side of the DNA molecule. And then this is going to be used as our template to make that make the instructions for the protein. So in our in this process, we use what's known as RNA polymerase. So keep in mind in DNA replication, we had DNA polymerase where we were bringing in those nucleotides for the DNA to copy it. Now we just have RNA polymerase, and I'm sure as you can guess, all it's going to do is bring in those RNA nucleotides in order to make that RNA molecule. One thing to keep in mind in transcription is that DNA gets unwound, because again, we have to unwind it, and then the RNA polymerase brings in the RNA nucleotides complementary to the DNA. Now, when we say complementary again, all we're talking about is that base pair that it matches up with. So if there is a T, a thymine in DNA, then we're going to get an adenine and an RNA. If there's an adenine in the DNA, then in the RNA, you want to say it would be thymine, but thymine is not in RNA. Thymine is only in DNA. So instead, we have a uracil, which hopefully you guys remember. So if there's an A in DNA, we get a uracil in RNA. And then if we have a guanine in DNA, we get a cytosine in RNA, and then a cytosine in DNA gives us a guanine in RNA. So here's our DNA strand. We'll call it ATCGCTACG. So take a minute and go ahead and transcribe that. Being able to do this step is very, very important in kind of understanding the whole picture and understanding how to go through this process. So give that a shot, see how it goes. I'll wait a second. Pause me because I'm only going to wait literally a second. Okay, so what you should have here is I'm just going to read the DNA and then I'll read what the RNA should be again right after. So DNA, ATCGCTACG, and the RNA is going to be UAGCGAUGC. Hopefully you guys got that. If not, please feel free to shoot me or Ms. Shantz an email so we can get some clarification, make sure you guys fully understand everything that's going on. And again, only genes are going to be copied here. And then once that mRNA is created, it's going to leave the nucleus and travel to the ribosome, which is out in the cytoplasm, so that way it can go through translation. So transcription, all it is writing down the message. Again, if you know how to go from DNA to RNA, just adding those complementary base pairs there, you're good to go so far. So now this is where things might get a little tricky here, the translation aspect. So now we're going from mRNA to our protein. So what's going to happen is that mRNA, that messenger, is going to be read or translated into the amino acid sequence. Now, keep in mind, mRNA is a nucleic acid. Proteins are, well, proteins. They don't speak the same language, so we have to translate it. We translate that using the help of our mRNA, our tRNA, and our rRNA. Keep in mind the rRNA is just that ribosome. You can see it back here, kind of in the back part of the picture. Our tRNA is this kind of prong rake looking thing going on, maybe a trident you could call it, that's coming in. It has, notice these 3 letters down here along with an amino acid on top, and then we're forming this polypeptide chain, poly meaning many, peptide being our amino acids, as it goes through, and then again our mRNA is just down here, that strand that has a sequence. What you'll notice is that this tRNA has a complementary set of nucleotides at the bottom that matches up with the mRNA strand. Okay. Now, again, when I say complementary, if we look over here at this last one, it has a uracil on the mRNA, it has an A for the tRNA, G, C, C, G, and so on. That's going to be very significant because that's what allows the tRNA to know which amino acid is needed in the sequence. Since structure equals function, we all know that. Each one of these different amino acids is going to give it a different function when in that protein. It's going to kind of determine how it folds and bends and how it's created at the end. So any difference in the amino acid sequence is going to cause a different protein to be made, which if we're looking for a specific protein, obviously we don't want mistakes to be made. We want to make sure we're getting those right amino acids. So what's happening? mRNA joins with the RNA. We form this molecule here, mRNA with our rRNA with our ribosome. tRNA then brings in those amino acids because of the codons. The codon is just a 3-base sequence on the mRNA, and the anticodon is on the tRNA. So mRNA holds the codon. So these 3 right here, wish I could draw, this CGU is a codon. It's complementary from the tRNA is the anticodon, kind of the opposite. Okay. Again, the codon and the anticodon match up, or the opposites rather, because that's going to tell it which amino acid to bring in. tRNA brings in that amino acid. As it moves down, it goes into the next one, and then so on and so forth. As it moves across, as this ribosome moves across the mRNA, reading it, it's going to detach that last tRNA, where it then can go back and get more amino acids or get another amino acid and bring it back in if we need another one of those amino acids. A peptide bond is going to form between amino acids. Hopefully you guys remember that from when we were talking about macromolecules. These bonds in here are peptide bonds, just a fancy term for the bond between amino acids. And then that ribosome shifts to the right, moving along. So I pointed the wrong way, but it's fine. Moving along as it's going to read that mRNA strand. And I'll move me. Protein, or the process rather, continues until a stop codon is reached. There are 3 stop codons and only 1 start codon. And that's going to be very important on this next page that I'm going to get to. We need to have a start codon. Okay, so the start codon is always going to be AUG. So we need an AUG on this mRNA in order to get it started. If it does not have that, it'll just keep skipping these letters here, or these nucleotides rather, until it gets a sequence with AUG, at which point it will start. So a single mutation in this can cause a lot of amino acids to be missed, and that'll be more evident later on when we talk about it a little bit more with mutations. So that start codon, AUG, every time, it always codes for alanine. That will always be our first amino acid. And then our stop codons, there are 3 potential ones. Once it hits that stop codon, it just stops and disassociates. There's no amino acid that correlates with the stop codon, which you shall see right here. So here are 3 stop codons, and then we're going to learn how to read this chart as well. We can translate the amino acid sequence using the genetic code. uses a 3-base sequence on the mRNA called the codon. So basically, if we go back here, this is showing us we have our mRNA strand. We know what our tRNA is going to be, the anticodon, but we don't know what amino acid it's going to bring in. We have to use this chart in order to figure out which amino acid it's bringing in. So, like I said, AUG is always the start codon. Every single time, without a doubt, AUG will be your start codon. So to read this chart here, we'll start with AUG as our starting point. First base in the codon, well, our codon is AUG. That's going to be the sequence on the mRNA. So we start with A. A is our first letter. So here's our A. Now we're stuck to these amino acids that are in this section here. Okay, just follow it across. We then go to our second base in the codon U. Okay, so A and U intersect in this box here. So now we're stuck just looking in this box, where we then have to go to our third base in the codon, which is AUG. So we have G as our third one. So we come down here, we find G, follow it across. All three of them intersect, A, U, and then G right here at the methionine. I should lie to you. I think I said alanine was the start. Correct me then. It should be methionine. Sorry about that. So AUG, MET is always going to be our starting point. Now, our stop codons, like I said, there's going to be 3 of them. So what would be the codon for our 3 stop codons? Take a second, try and figure it out. I'll give you guys literally a second, so pause the video if you want to try without me giving the answer in a second. Okay, so for stop codon number 1, and again, these might be in different orders, so if you have this as your third one, it doesn't make a difference, would be U, A, A. Okay, U, A intersects right here. Second stop codon could be U, A, G, come here, or U, G, A would be our third one. So now what I'd like you guys to do is take a second and tell me what amino acid or what 3 letters would correlate with a codon that is GCC. GCC. Okay, so G should start down here. We're stuck down here. C, G and C go into this one right here. So now no matter what the third letter is, it's going to be the same amino acid. Okay, so I think I said GCC, so it's alanine there. Now, what you'll notice is that there are possibilities of having the same amino acid with a different codon. Okay, there are many different codons that are potential here. I mean, if you want, you can figure out how many. 4 times 4 times 4, anybody know? 64. So there's 64 different combinations of codons. However, there is only, I think, around 20 amino acids here. So there's going to be obviously some different separations or different possibilities for the codons. Now, again, hopefully you all, I believe that's, yes. Here is the final chart. You should be able to read it using this one as well. You will not have to memorize these. You will always be given this chart to understand it. However, this one reading, it's a little different, but still pretty much the same. So we're going to start in here for our first one. So we'll go to AUG again. So we're down here in our first circle. Start with A. So now we're stuck in this lower left quadrant. Move me out of the way. And then we go to U. U is down here. So now in that lower left quadrant, the lower right, and then we just find G, which is right here, methionine, right there. And then you can just follow it that way as well. Now, notice things like proline. We have 4 different combinations there. And then we have a bunch of other ones. Alanine, right here, some combinations. We also have, valine, 4 different combinations there. So there are a bunch of different possibilities that you can get from those codons while you're reading this. So I believe that is the end. Yes. So hopefully this makes sense to you. We can also work backwards with it, which I can try and create a video. Yeah, I'll make a video of just working backwards with it and kind of figuring them out that way. So hopefully this makes sense. If not, stay tuned and you'll get a video that has me kind of working backwards and forwards from our proteins to our DNA and from DNA to our proteins. That way we can get some better practice with it. So thank you guys for watching and enjoy.